My name is Adrian Stanley. Recently, I found myself explaining to my friends Martin and Wesley how weed works in the brain. So I described to them the different effects that it has on different parts of your brain and why it does what it does. So I figured why not make a video about it, about that explanation, because I find myself explaining it a lot. All right. So basically, first thing I have to explain is the basic biology of the nervous system. So all things are made of cells. Brains are also made of cells, and they contain cells called neurons. These neurons, they receive messages from your other senses, and they interact with each other to form a complex network of neurons in your brain. And it's different neurons group together, and they do specific functions in your brain and in different parts of your body. So in the basic neuron function, the way they communicate with each other, when one neuron talks to another neuron, basically you have this is the basic structure of a neuron. It has a cell body. It has dendrites. These up here are called dendrites. And it has an axon, and it has an axon terminal. So in the axon terminal, there's a collection of these small vesicles called neurotransmitter vesicles. Basically, these chemicals whenever this cell wants to tell this cell to do something, it'll release chemicals from these vesicles. These chemicals are called neurotransmitters, and whenever it releases its neurotransmitters, the neurotransmitter will attach to proteins on the receptor of the cell that it's talking to. The cell it's talking to is called the postsynaptic cell. So the cell that it's talking to will have proteins on its membrane called receptors. These receptors will bind whatever neurotransmitter, and whenever those neurotransmitters bind to those receptors, it'll make the cell either excited and want to pass on a signal, or it'll make it not excited and make it not want to pass on a signal. So neurotransmitters can have either an inhibitory effect or an excitatory effect. All right. So basically, since you know that now, I can explain to you how cannabinoids work. Basically, you have cannabinoids in your body called endocannabinoids. These are basically these are molecules created by your body, and they act like they're your body's natural THC basically. So basically, what'll happen is whenever the presynaptic cell, which is a cell sending a lot of neurotransmitter signal, is attaching to when it's releasing a lot of neurotransmitter, those neurotransmitters bind to those receptors, and it'll increase activity of the cell. When the cell is really, really active, when it's getting lots and lots of stimulation from the presynaptic cell, then calcium channels on the membrane of the postsynaptic cell will open, and these calcium channels will come in. The calcium will come in through the calcium channels, and it'll activate certain enzymes located on the membrane of the postsynaptic cell. So basically, what these enzymes will do, they'll stimulate the production of endocannabinoids. So endocannabinoids like anandamide and arachidonoglycerol will be made in the intermembrane space upon activation of this enzyme here. And the endocannabinoids that are produced will then travel retrogradely, so they'll go from the postsynaptic cell to the presynaptic cell. So it'll travel retrogradely and bind to CB1 receptors. So these are cannabinoid receptors. So it'll bind to a cannabinoid receptor. And these cannabinoid receptors are attached to a G protein. So it's coupled to a G protein, and upon activation of this receptor, the G protein will interact with calcium channels on the presynaptic axon. So if these calcium channels are inhibited, then this neurotransmitter release can't happen, because they didn't show it in this illustration, but there's proteins that are attached to the membrane that are responsible for the vesicle fusion of these vesicles to the membrane to allow the release of the neurotransmitters. And those proteins are called SNAP. So you have this protein called SNAP25. It stands for synaptosomal associated protein. So these proteins basically, they require calcium, and upon calcium binding to them, they'll facilitate the vesicle fusion and allow for the neurotransmitter release. Now when the cannabinoid binds to its CB1 receptor and the G protein inhibits the calcium channel, then no calcium flows in to this cell, and as a result, if no calcium flows into the cell, then the SNAP protein can't activate, because it requires calcium. And since it can't activate, this part won't happen. The vesicle fusion and the release of the neurotransmitter doesn't happen. So basically, endocannabinoids act as negative feedback from if the postsynaptic cell is receiving too much stimulation from the presynaptic cell. All right. So that's the basic how endocannabinoids work. Now, basically, what happens is when you introduce THC into the neurons, basically what it's going to do is THC, which stands for delta-9 tetrahydrocannabinol, delta-9 THC, basically it's a receptor agonist, and what that means is it'll attach to and bind and activate CB1 receptors. So it's an agonist to CB1 receptors, and because of that, it won't, since the THC is directly attaching to the CB1 receptor, then you don't need the overstimulation of the postsynaptic cell in order for this to be active. So as a result, THC has an inhibitory effect on neurons. And this is important because if this cell, for example, was secreting neurotransmitters that make this cell excited, for example, if it was releasing glutamate, then this cell would as a result be less excited upon THC binding to the CB1 receptor, because now that this cell is inhibited, it's not releasing as much of its excitatory signal. This cell is going to be getting less of an excitatory signal than normal. And if this cell was an inhibitory cell, if its neurotransmitters had an inhibitory effect on the postsynaptic cell, then the THC binding to the CB1 receptor will result in this cell becoming more excited, because since this is not receiving as much of an inhibitory signal as it would normally. For example, if this cell was secreting a neurotransmitter like GABA. So now that you know that, you can understand how it influences your reward system. So basically, the reason why THC feels good is because of its influence on the reward system. So basically, in the reward system, you have called the mesolimbic pathway in your brain. Basically, you have this part of your brain called the ventral tegmental area, and this is one of the main areas that THC will bind in your reward system. The ventral tegmental area has neurons that synapse. They have axons that attach to neurons in the nucleus accumbens, and they have axons that attach to neurons in the prefrontal cortex. So the nucleus accumbens is associated with making you act out behaviors to want to seek reward, and the prefrontal cortex is your main area of decision making. It's your main thinking area of your brain. and the VTA has axonal projections that project there. So the ventral tegmental area has both dopaminergic neurons. Dopamine is a kind of neurotransmitter. It has both dopamine neurons and GABA neurons. GABA is an inhibitory neurotransmitter. All right. So knowing that, just focusing on the ventral tegmental area, nucleus accumbens, and prefrontal cortex, then you can see the basic way THC will affect the reward system. So in the ventral tegmental area, you have GABA neurons and you have dopaminergic neurons. These GABA neurons synapse on dopaminergic neurons within the ventral tegmental area. On the GABA neurons, these GABA neurons contain cannabinoid receptors. So whenever you take THC or just smoke weed, the THC will come in, it'll bind to the CB1 receptor. The CB1 receptor will inhibit the GABA neuron from secreting its normal GABA neurotransmitter. Since this cell isn't receiving the same GABA input, then it won't be as inhibited anymore. And as a result, it's going to be more excited. And because of this, it'll release more dopamine into the areas where it synapse. And the VTA has neurons that synapse in the nucleus accumbens and in the prefrontal cortex. So the increase of dopamine in the nucleus accumbens is what's associated with the pleasure behind smoking weed, the good feeling behind just reward. So in the prefrontal cortex, basically what the increase of dopamine in the prefrontal cortex will do is it'll induce this process called long-term potentiation. Long-term potentiation, basically, what it has to do with is it increases responsiveness of neurons in response to the same signals. So if, for example, LTP were to occur in the prefrontal cortex in areas that are associated with THC, so your reward system is in place to reinforce reward and make you want to do things that give you reward. So by inducing LTP in the prefrontal cortex in whatever gave you the feeling of reward to begin with, you'll think more about whatever it is that gave you the reward. So in this case, if you're smoking weed, the THC will bind here, and then as a result, dopamine will increase in the prefrontal cortex, and there'll be LTP in areas associated with the weed. So as a result, you'll think about weed more when you're not smoking or anything. And basically, when in your natural reward system, this will be in place for to make you want to be motivated to do things that naturally give you reward, like hobbies, exercise, and things of that nature. So basically, THC is bypassing this system by just binding here on the CB1 receptor. Basically, just one more thing: the way LTP is induced in the prefrontal cortex. I explained the LTP process in another one of my videos on NR2B. Yeah, so you guys can check that out if you want and see the exact specific mechanism behind LTP. But the general mechanism for dopaminergic neurons basically is when calcium or cyclic AMP is active within the cell, certain kinases will become active, and these kinases will phosphorylate. So these kinases will lead to long-term potentiation in these neurons, and as a result, those neurons will become more efficacious in their transmitting signals. Basically, what dopamine does, it'll bind to this D1R receptor, and this G protein coupled receptor will interact with adenylyl cyclase. Adenylyl cyclase will make cyclic AMP from ATP, and this will initiate the LTP process. Yeah, that's the basic what I wanted to explain, and I'll explain more in other videos. Alright, thank you.